All right, back for more. Day 50. We've been digging into nuclear receptors and all their complexities. Today, we're heading into a sort of, I guess you could say, a mysterious corner of this world, the orphan nuclear receptors.
Yeah, that's right. You could call them unsolved mysteries in the field, really, molecular biology. Huge potential for, breakthroughs in therapies, but we're still trying to find those keys to activate them, to find those missing ligands that bind and get them working.
Okay, so setting the stage here. We've touched on nuclear receptors before, right? These proteins in our cells bind to hormones, and then they regulate gene expression. But what makes these orphans, I guess, stand out from the rest of the family?
Well, it's like this. Unlike their well-studied counterparts, like the thyroid hormone receptor or the estrogen receptor, these orphans, well, we haven't found their activating ligands yet. Like having a lock, but no key.
So a real scientific treasure hunt, then. We know they're important, these receptors, but the treasure, the ligands to unlock their potential, still out there.
Exactly. And that's what makes them so exciting for drug discovery. Figure out how to activate these orphan receptors. Boom! New treatments for all sorts of diseases.
So which orphans are we looking at today? Any front runners in this molecular mystery?
Two that have been making waves, you could say, are Rev-Erb and ROR. Both of them are involved in some pretty fundamental processes. Circadian rhythms, our body's internal clock, and metabolism.
Whoa. Messing with our body clock and, how we burn energy. That's getting into some crucial stuff. I can see why these orphans are getting so much attention.
Absolutely. Think about it. Potential therapies for sleep disorders, metabolic diseases like diabetes, even some cancers. Game changers, potentially.
Okay, hooked. But how do scientists even start to unravel these mysteries? If we don't know what activates them, how do we even begin to think about, designing drugs to target them?
That's the challenge, right? It's pushing researchers to think outside the box. One way they're tackling it is with these large-scale screening techniques. Imagine taking a massive library of compounds and just throwing it at these receptors. See what sticks, what binds.
So it's like a molecular guess and check kind of thing, but on a massive scale, trying thousands of keys to see if one fits the lock.
Exactly. It's brute force in a way, but you can get some really valuable clues. And then there's computational modeling. Researchers use computers to predict how different molecules might fit into the receptor's structure. Virtual puzzle, finding that missing piece that completes the orphan receptor.
So high-throughput experimentation combined with some serious computer modeling, that's a cool blend of cutting-edge tech and good old-fashioned detective work.
That's a great way to put it. But it gets more complex, Even if you find a compound that binds, it doesn't necessarily mean you understand how to actually control its activity.
Oh, wait, what do you mean? Can you give me an example?
Sure. Let's take two receptors in heart muscle cells. The beta-1 adrenergic receptor and the EP4 receptor. They both trigger the production of a molecule called SEMA-P, but their effects on the heart, totally different. Beta-1 increases the heart's contractility, EP4 doesn't.
So it's not just about, flipping a switch. It's about understanding the nuances of how that switch connects to all the different cellular machinery.
Exactly. And that's where these kinase anchoring proteins, or AKPs, come in. They act like cellular zip codes, guiding the activity of protein kinase A to specific spots within the cell.
Interesting. So you're thinking that understanding how AKPs guide protein kinase A activity could give us clues about how ligands might bind to and activate these orphan receptors.
It's definitely a promising avenue to explore. Both processes rely on really specific binding domains and structural features. For example, AKPs have this type of helix, and an amphipathic helix, and it's crucial for how they interact with protein kinase A.
I see. So you're saying maybe these orphan receptors have similar structural features that their ligands are latching onto. And if we understand those features, it could help us identify the missing ligands.
Exactly. And that brings us to another approach researchers are using to crack the orphan receptor code. Studying receptors where we already have the key.
So we're taking a detour, learning from a solved case before we tackle those cold cases, are orphans.
Precisely. By studying how known ligands bind to their receptors, we can get insights that might help us understand how ligands might bind to these orphan receptors. And a great example of this is the thyroid hormone receptor, or TR, right? Thyroid hormone, it's essential, regulates our metabolism, growth, development, the whole 9 yards. And we've learned a ton about how it interacts with its receptor, right down to the molecular level.
Okay, so break it down for me. What's so special about how that thyroid hormone binds? What's the lesson here?
Well, there's this helix in the TR ligand binding domain called helix 12, and when that thyroid hormone binds, it It causes a change in this helix. Like a key turning a lock sets off a whole chain reaction inside the cell.
So Helix 12 is like the VIP, the key to activating the thyroid hormone receptor. And you're thinking maybe our orphans, they have similar, uh, I guess, structural features that do the ligand binding thing.
Exactly. See, there are these motifs, helix turn helix motifs, in protein kinase, a regulatory subunit. And they're essential for docking onto, you remember those AKAPs we were talking about? And those motifs, they kind of resemble helix 12 and TRs.
Ah, so maybe those helix turn helix motifs in our orphans, they could be like
Yeah.
signposts pointing to how their ligands are interacting.
Yeah.
It's pretty cool. Really shows how studying a, well-understood system like the thyroid hormone receptor can be a roadmap for these uncharted orphan receptor territories.
Exactly. It's all about connecting those dots, seeing the patterns in the molecular world. And remember, we're talking about therapies here, revolutionary ones. Unlocking these orphans could totally change the game for medicine.
Right. Okay, we've talked about the possibilities, but let's get real for a sec. It can't all be easy, right? I mean, this is cutting-edge stuff. I'm sure there are roadblocks all over the place. What kind of challenges are researchers up against in this field?
Yeah, no, you're absolutely right. It's complex, it's challenging. One of the biggest things is just how much we still need to figure out. We got to characterize these orphan receptors better, understand exactly what they do, how they work in different tissues, what other proteins they're talking to, the whole picture.
So we've got this toolbox full of potentially awesome tools, but we're still kind of figuring out which one does what and how to use them right.
That's a good way to put it. And it gets even more complex. These receptors, they might have activities that go beyond just regulate gene transcription, what we call non-genomic activities.
Okay, now that's got me curious. Non-genomic activities. What's that about? It's like they've got secret side hustles besides turning genes on and off.
Think of it this way. They might be moonlighting. They might be interacting with other proteins or messing with different signaling pathways right there in the cytoplasm. It adds a whole other layer to what they can do.
So like these orphan receptors might have hidden talents we haven't even uncovered yet. No wonder this research is so tough, but also so exciting. It's like exploring a whole new continent in molecular biology.
Exactly. And that's why keeping the research going and having collaborations across disciplines, that's crucial. Bringing together the best from, molecular biology, pharmacology, computational biology, and more, that's how we tackle these challenges.
It's like assembling a dream team of scientific detectives to crack the case, right? These orphan receptors. Okay, so we've talked about some of the strategies like the large-scale screening, computational modeling, studying those well-understood receptors like TR. Anything else scientists are looking into?
Well, one pretty interesting approach is looking at what happens when these receptors, malfunction. Sometimes you can learn a lot from the broken versions. Like think about those mutations in the TR gene that happen in patients with resistance to thyroid hormone or RTH.
Right, so by studying how these receptors are acting, we get clues about the normal way TRs work. It's like taking apart a broken clock to figure out how the working one, how it keeps time.
That's a perfect analogy. And there's another example of this, the Rev-Erb oncogene. It's a mutant form of Treg found in this virus, causes cancer in birds.
Oh, wow. So this viral gene, it's like it's hijacked a normal cell function, twisted it for its own evil purposes. And if we understand how it does that, we learn more about how the neuron receptor works.
Precisely. See, by understanding how Rev-Erb messes with cellular processes, we get insights into how normal Treg functions, how it contributes to cell growth and differentiation. And the same approach, it applies to our orphans too.
So even though we're still looking for those keys, for these orphan receptors, we can still learn from situations where their activity is disrupted. It's all about, piecing together that puzzle, using every clue we can find.
Yeah, it's a fascinating puzzle, no doubt about it. And as we keep digging deeper, we're not just, uncovering the secrets of these receptors, but we're also getting a better understanding of how our cells work, like at the most basic level.
It's wild to think about all that therapeutic potential just sitting there, hidden in these receptors. Like we're standing on the edge of a whole new continent in medicine, just waiting to be explored.
That's a great way to put it. And with all the research going on, the new ways of thinking and people working together from different fields, I'm sure we'll find those keys to unlock these mysteries.
It's mind-blowing to think these orphans could change everything about how we treat so many diseases. We're talking about potentially huge changes in how we approach things like sleep disorders, metabolic diseases, even cancer. Makes you realize how much potential there is in this whole area.
Absolutely, and it shows just how important it is to keep investing in basic science research. These discoveries, they don't just happen overnight. You need patience, you need to keep at it, and you gotta be okay with exploring the unknown.
You've definitely left me with a sense of awe. And I'm even more curious about the intricate stuff happening in our cells and the incredible potential of scientific discovery. So
Yeah.
for everyone listening, what's your takeaway from our deep dive into these orphan nuclear receptors?
Well, I hope everyone's walking away with a sense of just how complex, but also how beautiful the molecular world is. And maybe a new appreciation for those scientists working so hard to figure these mysteries out, how dedicated and creative they are.
I totally agree. And if you're as fascinated by these orphan receptors as we are, do some research yourself. There's so much information out there, and who knows, maybe you'll end up being one of the people unraveling these mysteries. Until next time, keep exploring and stay curious.